Once again, today we're going to be talking about super resolution microscopy systems. And for those of you who may not work a lot with microscopy or may not work particularly with super resolution, that simply means being able to see fine structures that otherwise laser scanning microscopy with light wouldn't be able to define. So this is an example image of comparing a wide field microscope where our illumination goes across the entire plane on a structure called vimentin. Here is the same region where we do a confocal scan. We clean up a lot of these fibers and structures, but we still have quite a bit of blurriness that hopefully what you can tell on your screen, maybe better than a projector, is that there's a fine mesh of these structures here that we're able to actually resolve through super resolution technologies. And particularly with today's focus, we're going to be discussing super resolution related to fluorescence microscopy. Fluorescence as a physical phenomenon is one of the most flexible and dynamic ways of being able to generate contrast and study biology. Starting with subcellular structures at the nanometric scale, we can see how individual proteins and mitochondria, organelles, interact within a single cellular environment. But we can also understand how those cells form an ecosystem in order to discern differences in metabolic states and different diseases, and also even serve on a macroscopic scale to aid in surgical intervention. So from nanometer to centimeter, fluorescence is a key tool that allows people to not only understand biology, but to treat disease and to make huge developmental strides in medicine. And to be able to see in super resolution has been a recent, in terms of the history of microscopy, development. For hundreds of years, we've had microscopes available to us to see things that we couldn't detect with the human eye. And in particular, though, these structures that you see here on the confocal image of blobs are called nucleopore complexes. And the nucleopore complex, or NPC, has been a very, very hot topic in super resolution microscopy because super resolution, for the first time, aside from electron microscopes, was able to really redefine and pinpoint this structure. And the NPC is super important because it represents the unique point of entry and for two-way exchange of different proteins and molecules and viruses, particularly in and out of the nucleus. And so we can hopefully appreciate why being able to see where a virus might enter and what things it might interact with at a very small scale, particularly in light in the last 2 years, being able to see small things helps drive medicine and aid in treatment. And so before we get into more of these super resolution images, their uses, and how we can achieve that, we're going to talk in what might be largely a review for many of you, a bit about light microscopy and fluorescence itself. So fluorescence is going to be the key principal phenomenon that allows us to generate the contrast we see in cells. It is a physical phenomenon that occurs when we have a molecule that almost acts in resonance. So it can receive light, and when it gets a wavelength that is special for that molecule, it gets to an excited state. And almost simultaneously, that excited molecule kicks off energy. And when it loses energy, it emits light back off. Now that emitted light is at a lower energy than it was previously because of conservation of energy. And that wavelength is going to be much longer than the first wavelength that we excited it with. So this difference here is called the Stokes shift, and this is what gives us contrast between the light that we excite with and the light that we image with. And essentially, we exploit that Stokes shift in the standard fluorescence or standard wide field microscope, where we send light in, hits the sample all across the entire plane, everything in it, not just in the focal plane, and comes back up to a camera. And so this was important because when we want to study cells and we want to see things that we couldn't otherwise detect with the human eye, we need to be able to label individual things because they are actually very clear. Our cells, human cells, are clear to the naked eye. And so we can see then in color and study dynamic processes. And so the advent of the wide field fluorescence microscope had really, really changed a lot of what biology could do. for the first time, people were able to visualize these processes of cell division and the different phases of mitosis. But as important as it's been, there are still some limitations that plague fluorescence imaging, and particularly the wide field microscope. It's a powerful tool, but the light that we shine on a sample can over time dim the signal that we see back from the labeled structure. This is called photobleaching, so the light on the fluorophore dimming over time. If we use really intense light, we can also have the problem of killing the cells. As we likely know, if we stay outside in the sun for a long time and for a lot of frequency over our life, ultraviolet rays can rip through our DNA and cause damage. So phototoxicity is a problem with fluorescence microscopy. And a big theme that we're going to see that is the main driver for the advent of super resolution microscopy is this physical property of light called diffraction. Light spreads through all media, and when it spreads, the bigger it spreads, it really limits our resolution. And finally, in the wide field microscope, as I mentioned on the previous slide, the light floods the whole plane. So we're not just seeing things that are in the immediate focus, but sometimes we also are getting light coming from the same structures, but they're just in a different plane. And that can reduce our contrast and make things look a little more blurry, or it'd be hard to identify fine structures as well. So the confocal microscope was able to really significantly address that final point. So essentially, the way the confocal works is instead of flooding the whole area with light at once, we have a laser that we pass through a first pinhole before deflecting it onto mirrors. These mirrors are then going to scan across in a snake-like pattern that we call raster scanning on the cell. Then what we get back is emitted light, again, a longer wavelength, a different color, and we're going to pass that through a second pinhole that's aligned in such a way that they are confocal. And that's going to help us greatly reduce the out of focus light to improve our SNR. It also is going to enable deeper sectioning through tissues, so we can actually take pictures several microns deep within the sample. But importantly, we do have a diffraction limited challenge here. So even though we've improved from a huge flooded lamp of light across the sample and we've now pinpointed a laser, this spot is called the area spot. This is what we scan with. The red represents the most intense area, but the intensity drops off somewhat uniformly in this three-dimensional Gaussian shape. So that we call this the area spot, and we can only see structures that are as small or as big as the diameter of this spot. That's proportional to the wavelength of light that we use to excite with, and also some properties of our objective, like the numerical aperture, which corresponds to the angles of acceptance that we can have. So this is going to become very important for being able to see things small. So we can only see as small as that diameter, roughly maybe divided by 2, or we can only detect 2 nearest neighbor objects if they're at least this distance apart. And although that technology covers a great deal of the spectrum on these different spatial scales, from organelles all the way up to whole animals for imaging, we still hit a limit here with anything below the diffraction limit, which is between 250 and 200 nanometers, depending on your confocal microscope, that anything smaller than that, we're going to have a hard time seeing. So with that in mind, this is why we need super resolution imaging. So we need to develop some techniques and ways to work around this diffraction limit challenge. And there's 3 ways that we can do this. So 1, we can think about how we could maybe make that scan spot smaller. Another way is maybe instead of, if we think of a confocal area spot, like a whole wide field image of flooded light, maybe can we pattern the light that our sample sees so we can separate nearest neighbors? And the final way is maybe we can think about changing the sample itself. So if the light itself remains the same, like a wide field microscope, perhaps we can induce areas where we can turn single molecules of fluorophores on while others are off at the same time. So with these 3 approaches in mind, we're going to talk about the different technologies that exist, but I want us to keep in mind that there are some challenges to this. And each one of those three methods has its own unique issues. So one big limitation is that to do some super resolution techniques, the imaging itself can take a very, very long time. Some techniques also require pretty intense lasers to induce the phenomenon to get below the diffraction limit, and that can cause damage to our sample. Some of those techniques, particularly the one that relies on getting individual molecules to blink, are going to require particular sample preparation. It's not just what I would normally do to put my sample under a confocal microscope. I might need to do some chemistry or play around with the different fluorophores, and maybe not all will work. As we'll see, that's the case. We also think about how expensive and accessible the equipment can be, because some of these technologies can be quite expensive, and that's why we don't see them everywhere. And importantly, in any case, like with any type of imaging, we can be prone to artifacts. Some techniques more than others, some techniques differently than others, but these are all issues that we need to keep in mind when we understand what we're about to talk about next. So we're going to summarize some of these things in a pentagon diagram, and these are going to be the 5 points that we consider. So the 5 core aspects that we'll consider are, first and foremost, the spatial resolution. That is our ability to see very small objects. The second is going to be how fast we can see events, or particularly short-lived events. The temporal resolution can be imaged quickly. The other consideration is now in depth. Can we actually practically get super resolved images deeper into the sample, or are we limited to a very thin film? Is the technique compatible with live cells? Because a big thing that confocal users use their microscope for is to be able to see real-time dynamic processes to study disease. And finally, but not least, is the simplicity. We're going to weigh the cost and potentially the time and expertise needed to prepare samples and actually achieve the promised super resolution. And so now we'll talk a bit about those techniques that exist and how we can go beyond the diffraction limit. So one of the first techniques that came about in the last 20 years is called stimulated emission depletion microscopy, or STED. STED is going to borrow this scanning analogy from confocal, where we have a beam that raster scans, but instead of a single excitation beam, this airy spot here, there will be a co-aligned secondary beam of a different wavelength that's effectively going to cancel out and erase all but this remaining small spot. So the effective point spread function, a limit to the resolution of the system, is going to be much smaller than we'd otherwise see with the airy spot. So this technique has a lot of advantages. We can get really great resolution directly, close to around 50 nanometers. This can be done in up to 2 colors, 2 different colors of excitation. Some systems and technologies are working on getting more. And another way you can add additional channels of contrast is by combining STED with another technique. For example, one that not can only discriminate in color, but also in time, if you use 2 fluorophores of the same wavelength, but one has a longer fluorescence lifetime, for example. The post-processing that one needs to do is quite different and much more simple, because really we've created a much smaller scan point. But this technique can be subject to some particular challenges and limitations. The intense STED depletion beam, this donut here, can over time photo bleach or be phototoxic to your sample. And as we do this scanning, it's going to require stable dyes, and we also need to consider the time, because these 2 beams are going to need to scan on a very, very fine grid. So they might take a bit longer to acquire the image. It's a bit more of a depth-limited technique, and it can be difficult for live cells. So we'll get better resolution, of course, if we look into a fixed cell, because it won't be moving. Finally, a conference I was recently at in Spain, we had a whole session on discussing both STED and another technique, STORM, and there are some real challenges to the necessary alignment. These 2 beams need to be perfectly aligned in order to get the accomplished effect. So this does take some expertise and someone that really knows quite a bit about handling their lasers. Another way that we can do super resolution imaging is by increasing the size of what the camera sees. So the technology by Confocal NL, based in the Netherlands, called the rescan microscope, they use a geometry really similar to a confocal microscope, but there's a second set of galvanometric mirrors that actually scan again or rescan, and they enlarge the image on the camera. So the increase in resolution comes from this deflection beam onto the camera. This technique is versatile. It allows you to work with live cells. You can do multiple colors. Your resolution, you can on average get 170 nanometers. That's still below that 200 nanometer level. And with more deconvolution in your image processing, you can get up to 140. No special sample preparation is required, and it's both fast and simple to use. However, the setup is a little more complex than a traditional confocal system. And in order to get to that 140 nanometer resolution, the algorithm is going to have to go through deconvolution, which some people just may need to consider if you need to keep a lot more information in your project. And there's kind of a ceiling threshold to the resolution that you can actually obtain. So 140 is kind of where this technology has been sitting at. So another way that borrows a bit, again, from the confocal scanning is the ARRI scan. So this uses another confocal type analogy, but instead of using a pinhole, there is a hexagonal shaped detector. And so this detector allows us to capture a lot more light. So we're making a lot more use of the signal that we are getting from the total amount of images. So this is very compatible with live cell imaging. You can use multiple colors, get up to 140 nanometers of lateral resolution. No special sample preparation is desired. It's simple to use, well established, and it's also fast as well. However, something to consider is that the ARRI scan will only be compatible with a Zeiss microscope. So the user definitely needs to have the same brand of microscope to be coupled with this. Deconvolution is going to be the best bet for getting down to the best resolution. And just so the user knows that you do need to go through a calibration step prior to acquiring your images. Now, if we take a step back and look at more refined, getting better spatial resolution for super resolution fluorescence, the top of the top here is going to be these stochastic techniques: photoactivation localization microscopy, PALM, and stochastic optimization reconstruction microscopy, or STORM. These offer the highest possible resolution. And the way that they work is we put a wide field like a wide field analogy of light on the sample, and the fluorophores are very special. So you need to use particular fluorophores that we can over time induce blinking. And by syncing the camera with the blinking events, we can take thousands, if not tens of thousands of pictures, and we're able to capture and reconstruct each single molecule over time. You can do this up to 20 nanometers, possibly even a little better, a little worse, depending on the sample. You can do it in multiple colors, and it uses relatively low illumination power to achieve this, which is a good thing because you need to do this many, many times over. So the biggest limiting factor with this is, well, a couple things, but in particular, we must use specific fluorophores. You can't just use anything you would normally use. You need to have one that will blink. Usually that means something that blinks is going to need a buffer, and many of the existing buffers will kill live cells. So it can be very impractical to do these kinds of techniques on live cells. There is definitely people working on it, but it's just something to consider. The best resolution that we can possibly get, though, is going to be much more limited to a very shallow depth, and this is going to take a long time to acquire. In terms of really nailing down the sample preparation for techniques like PALM and STORM, in order to get the fluorophore to label the structure, you need the right chemistry of adjusting the permeability of your cell, which can change based on cell type or structure type. It can take people a long, long time to really nail down the sample preparation. It's a big thing to consider, but the benefit of spatial resolution is certainly there. So another technique, if we go back to thinking about what we can do with our illumination, is structured illumination microscopy, whereby we pattern light in a barcode-like pattern. So here we're sending in light that's alternating between on, off, etc. And we can rotate that across the image and really detect fine structural details of the edges of the structures of interest. It's certainly compatible with live cell imaging. You can do it in multiple color channels, no special sample preparation, easy and fast. But we do need sensitive cameras to pick up these details. Phototoxicity, bleaching can still be an issue, so we want to make sure to choose stable fluorophores. This technique historically has been prone to some artifacts due to the way that the reconstruction is done. And if we have a lot of background signals, so really dense fluorophores, it can be especially prone to artifacts. And to get very, very good resolution, we can be a bit more limited in how thick the sample can be. And finally, last but not least, we talked about all these other techniques and kind of saved this last one because it required a little bit of seeing a few combinations of things to appreciate what conical diffraction microscopy does. So I'll talk a bit about how this works, but this last technique that's called Live-CODUM, it's a T-Light product, just being forthright, is going to combine the strengths of 3 different elements that are foundations of other super resolution techniques. So it's going to retain the geometry of a confocal microscope scanning system, but instead of just a single area beam, the pattern itself is going to have a structure to it. And so in that sense, that structure, it's going to have a region of no light being directly delivered to the sample. So we're actually directly optimizing our point spread function. And so the way that conical diffraction microscopy works is this whole pattern is going to look very similar to, and the schematic is very similar to a confocal microscope. We take laser light that we pass through a conical diffraction beam shaper. Conical diffraction occurs in media, in our case a biaxial crystal, where this crystal has a property of each x, y, and z having a unique index of refraction. And when we pass polarized light through that beam shaper, we can produce patterns of light and structure them. Then we can take those structured patterns of light and scan them across the sample. So now instead of us seeing this airy spot, which you can certainly produce with a conical diffraction beam shaper, we're actually going to see this 2 lobes that we call half moons. And using voltage controlled Pockle cells, you can very quickly rotate them. And so for each point, we scan with this shape instead of this one. And instead of a detector, we actually have a camera. So all the light signal is coming directly from the sample. So we're going to be able to take that information and put it directly into a reconstruction algorithm. Okay, so at this point, we've seen lots of different techniques. We've seen that fluorescence is a powerful tool, that confocal microscopy is still our big workhorse, but there's a lot of ways we need to work around this diffraction limit. When we try to organize our thoughts about how to look at these different super resolution techniques, I want to make this more simple and think of 2 different goals and outcomes based on super resolution imaging. So one, very important, is choosing a system or a technology that aims at getting the best resolution. So these are going to be our techniques and technologies and products that really focus on optimizing this part of our pentagon. So these techniques are going to definitely get us the best spatial resolution available, but they're often expensive and they can involve some complex considerations in terms of alignment, setup, and sample prep. Typically, because of this complexity, it requires a team of people that can devote time to becoming experts in this. And so these products typically are limited to bigger platforms, and they can be much more expensive than other solutions. And they're great. However, like I said right before this talk even began, all biological questions don't necessarily need the strongest resolution that the market can offer. Not everybody needs to see these individual molecules, and some structures don't have any refined things happening below the diffraction limit. So the goal of like where these types of products are headed is to try to match what electron microscopy has typically done for the field. So transmission electron microscopy is kind of this holy grail of spatial resolution. But that's kind of it. It did truly wonders for the biochemistry and biology world and history, but it does have a lot of drawbacks, including how expensive it is. In fact, the entire microscopy market is really dominated by how expensive these products can be. They require stringent sample preparation and lots of imaging time, and definitely the expertise. So working towards this goal in mind, there are projects and products that are working towards getting to this standard. And by this, I mean like single nanometer level, but using fluorescence and doing it in live cells. So some projects include the MinFlux technology, but also a product that we're working on called dark tracking. So these techniques are aiming for single nanometer level. The second goal of super resolution might be in a different direction, but that's going to be towards a more balanced acquisition tool. So these are systems that are going to be more all-purpose, that have a very flexible range of use cases. They're going to have lower resolution than those ultra-high 50 to 20 nanometer technologies, but they will make up for it maybe in terms of depth penetration, speed, and compatibility and flexibility. They may also be more customizable and of interest to people with more unique setups. The sample prep, absolutely easier, very little, if any, deviation from what you do for Confocal. And we have a big variety here. These shapes look quite a bit more similar than the previous ones in terms of the strengths, but it's important to know that some have strengths where others have weaknesses, and we like variety, as we want technology to be as diverse as the biological questions that we have to answer. And so too, just as I mentioned, not everyone needs single nanometer level. These techniques for a lot of people are going to be very sufficient, if not more so, in terms of spatial resolution. So here the end goal is kind of creation of a super confocal device. So based on that gold standard of confocal microscopy, we want to drive towards having a flexible solution, but with greater resolution, and we consider the different strengths and benefits. All right, so we're coming into the last two sections here to talk about how now we can use super resolution, apply it in action and in biology. So here what we're seeing is an example of using structured illumination. So here, this research group wanted to study the cell contractility. At the time this paper was published in 2014, the authors reported that there were very little tools available to actually characterize cell structure in terms of migration and how it reacts to different contractile environments, because cells can change their shape according to what kind of media they're in, but also when it comes to disease. So what we're going to see in this video here is actually the front forward region of a cell that's actually going to be kind of crawling and inching forward. So these green regions of myosin are actually moving forward on these red actin threads, and SIM allowed people to see that. Also to here, it allowed them to quantify the direction of these actin filaments according to the different heights. So here the color code is corresponding to height or depth of the sample. Now, another thing too, along the lines of structural biology, is not just understanding how individual structures themselves look, but also how do they interact with each other. So in this particular paper, what we're seeing here are some green proteins. They are in fact punctate that correspond to secretory bodies that were suspected to localize near or on, in purple here, microtubules. So here in these wide field images, we can see the structures, but really we don't get a lot of detail. Now these researchers compared both what SIM could give them and what single molecule localization microscopy using a ground state depletion system, which is very similar to STORM. So on the SIM image, they were able to see that it appeared that the the green Senex protein did locate on the microtubule, but some blurring indicated that there might be structures beneath the limit of what SIM could give. So here, in fact, we can appreciate these punctate structures localizing around the microtubule and also having unique shapes that we're not able to as finely characterize as with SIM. And so here, once again, we're seeing just individual microtubules. This paper came out 10 years before the previous one, but kind of if there's anything to take away from the images you've looked at today, it's going to be comparing E and F here. So here in E, with conventional wide field microscopy, we have a really thick fiber that looks like it should be a microtubule. So we're looking at this region here. This appears to be thick, but once we get down to doing single molecule localization in STORM, we actually see that this is 2. Likewise, here we have 2 microtubules. So this previously, when we only had wide field at our disposal, we would never be able to discern these individual fibers. And so to wrap things up, keeping this in mind of all the things that we can see, I'm gonna show you a bit about what we can achieve with not just STORM or SIM, but this unique technology that T-Light offers with the with the conical diffraction microscopy. So here, that same image you just looked at, those microtubules, we saw that they had a spotty-like structure using the STORM technique. What we can see here is actually the same punctate labeling. So we're using an antibody labeling, and this is not an artifact, but it is indeed a real observation of the discrete nature of antibody labeling of a microtubule. So what we can achieve is really refining not only the contrast, but the resolution. The Live-CODUM system is actually 3-in-1, so it localizes in wide field. You can perform confocal scanning and then achieve the super resolution image of your cells in multiple colors at the same time. And the way that this sort of works is the optics which produce these images here are all contained within this box that sits on, a tabletop connected to any microscope. As long as your microscope has an available side port, you can attach the Live-CODUM system on it. So within it contains the conical diffraction beam shaping. So we have our laser. It goes into that biaxial crystal after we polarize the light. And then we can use the other polarizers to produce these 2 half-moon patterns. Then those half-moons are scanned for each point in the sample, like a confocal microscope. We directly sample pixel by pixel locally, like exactly locally, how each distribution interacts with each part of the sample. And from there, since we're taking information from a camera, we can extract the higher frequency, spatial frequency information using maximum likelihood algorithms and produce a super resolved image. And so what you can get is kind of in that nice in-between super resolution resolving power. So we have up to 90 nanometer lateral resolution, and we've validated it in these DNA origami samples. So these 2 fluorophores are separated on DNA origami by 90 nanometers. And here too, we can section through a tissue, and axial resolution is then going to be identical to confocal. And so here, this is a 60 by 60 micron image. And this insert here is showing us that what we're able to see in this green channel, hopefully you can detect this fluorescent label labels only the outer mitochondrial membrane. So we can actually resolve inside the crypts of mitochondria in these cells. And so just some of the specifications, here it is connected to another microscope on a different side, but we can resolve 90 nanometers. We scan with exposure rates that can vary from 40 to 100 microseconds, even longer, of course, but here we can scan down to 40 microseconds per microimage, which means that for a 10 by 10 micron image, it'll take about 1.5 to 2 seconds. The wide field that we've attached onto the back port of this microscope, we use a wide field of view that we image with the camera that's inside the box of 90 by 90 microns. And then you can draw a region of interest to do your super resolution imaging to 60 by 60 microns. Now, this is going to vary a little bit based on the computer, but it's a flexible thing. We have off the table coming in through the back here on this particular model, we use 4 different excitation channels, which correspond to the most frequently used in biology. But the phenomenon that produces the conical diffraction is actually achromatic. So if a researcher said, I want this one, this one, this one, you can retrofit the device to be combined with any visible or near infrared wavelength combination. So it's very flexible in that way. The studies that were done to look at phototoxicity, this system uses really, really low light power. We rely on a linear fluorescence response in the sample itself, and we don't need a lot of light. So incident power on the sample, super small. So phototoxicity, there's almost none. It's like in the single microjoules of energy on the sample. It's definitely compatible with live cell imaging and incubator fittings, and the tentative tissue penetration depth that we've seen in an older model of this system was about as good as Confocal, so being able to scan and achieve super resolution down to 0.5 millimeter. And so to wrap up our discussion today and hopefully kind of review some of the big points that we talked about, the first being that fluorescence is a super diverse and powerful tool that allows researchers to label and examine living structures and cells. Confocal microscopy has been and remains the gold standard in terms of its versatility for biological imaging, being able to penetrate samples with depth, and also to do so quickly. However, there's a whole world of questions and more questions that continue to be opened up through the doors of super resolution microscopy, because the limit of spatial resolution led to this need to develop powerful tools to allow us to see these smaller sub-diffraction structures. And just as biology has a diverse breadth of questions, this means that we might need unique solutions, so that maybe the highest spatial resolution, not everyone needs to do electron microscopy the same way that not everyone may need to do more stochastic techniques like Palm or Storm. There's a trade-off between the five elements that we presented today that the user wants to consider. And fortunately, we have many different tools available that can satisfy the goal of both high resolution and flexible acquisition needs. At this point, I would love to take some questions if you have them, and that's all I have for you today.